going again, and we're going to have the Crystal be our first presentation, and then our second presentation will be by Kevin Hicks, who runs our Metabolite Protein Discovery Core.
Can everyone hear me okay?
Yes. One more thing, Crystal, let me just pop in and say, for those of you who are on, you can interrupt and ask questions. You can populate questions in the chat. But during the presentation, I would ask you to mute yourselves and then just unmute yourself when you have a question. And I'll go through and mute you if you don't mute yourself so that we can not have those interruptions. Thanks so much for everybody and participating.
All right, great. Thanks everyone who has joined us this morning. So my name's Crystal, and I've been director of the Mutation Generation Detection Core for the past 4 years or so. And in 2021, I also became the co-director of the transgenic gene targeting mouse facility. So today I'm going to focus my talk on the CRISPR technologies we're using to make genetically modified mice. But first, I'm going to give a brief overview of kind of the core structure and an overview of all the services both cores offer. So how our core service workflow kind of goes is I receive user requests, and then in my core, I work towards, in the Mutation Generation Detection Core, I come up with project design, editing, reagents, or sometimes people just come to me for genotyping or sequencing data. So I take in mind what type of edit the user needs to make and what model, and produce the most optimal reagents. And then those reagents, I can either, in some instances, I give them back to the user for them to use in their lab. And this is really important for people that have non-standard model organisms or cells with specialized conditions. Or I work with other cores, so with the transgenic gene targeting mouse facility, which I am part of, to generate the mice. So this is where we do the injections and electroporation and the mouse husbandry. I also work with Dr. Bilo in the drug discovery core to generate immortalized cell lines, and with Dr. Colin McGuire and the cellular translational research core, and he works with iPSCs, and we can also work with some primary cells. And when I'm doing work with these cores, so they introduce the CRISPR reagents to the models and then give the MGD core genomic DNA samples for analysis. And then once we have a characterized line or organism, we give that back to the user in the end. And for those users that do the work in their own lab, we're also there for support if they're having issues with introducing the reagent or analyzing, we can support them as well. So in the MGD core, our main services are CRISPR, Cas consulting, design, and reagent generation. We do in vivo validation of CRISPR, cutting efficiency, generation of knockout cell and animal models, and also knock-in animal models. We have genotyping services, sequence verification of genome edits, and custom cloning of vectors. So the kind of reagents we provide, so we mostly provide synthetic CRISPRs because these show the highest efficiency. And we also have high fidelity Cas9 protein. So users can use this CRISPR RNP technology that works best for in most instances, but we also have CRISPR Cas plasmids. So you can do, I don't know why I did that, excuse me. AAV CRISPR, CRISPR interference and activation also is in a plasmid format. And when required, we can get unique plasmids for non-standard models. And then Also for CRISPR-Cas editing, we produce homology-directed repair templates. So these include short modified oligo donors for making single base pair chains or changes or inserting small protein tags. We can make long single-stranded DNA donors for targets up to 2 kb insertions, and double-stranded DNA targets. I have some timing going on for even longer inserts.
Crystal,
Yeah.
this is Diane. Will you go back one slide? And so what's the efficiency of the short inserts, like a tag, versus a longer tag?
Yeah, I'll go for this more in my talk, but the short inserts are much more efficient. So we've seen instances where about 50% of the founder animals show insertion. And I'll go for this more later too, but where then the founder animal actually looks like every allele was edited. Like if you sequence it, it'll show 90 to 100% editing. So very efficient. And then for the long single-stranded donor, we can see pretty high efficiency with like 36% insertion, or so 36% of animals.
That's pretty good.
But they're much more mosaic. And then the double-stranded's low efficiency, like less than 10.
Thank you.
So then in the MGD core, we also have genotyping services, including high resolution melt analysis to detect small nucleotide changes. And this is what we use currently to validate CRISPR reagents. And then we do traditional PCR genotyping and also clonal sequence verification of induced edits.
Can I ask a question here already?
Yes.
So I'm kind of in here for personal reasons, as anybody would be, right? You have personal interest, but right, we used your services to do CRISPR cell lines, which is great.
Yeah.
And except, you know, had some issues with heterozygous clones. And so we used the two heterozygous phenotypes that we've got, and we were trying to move forward, and we're getting very different phenotypes. And I'm just wondering, is there, when you validate these or go back, do we have that sequencing information to see what's different between the two?
So yours was, so sequencing info, usually when we're making an insert, we'll sequence that. So yours was a deletion. So you can get that data from traditional PCRs. But if we could revisit your lines and certainly sequence them. And I remember your project very well because the clones wouldn't grow well. So then Dr. Lowe, he had to do serial dilution. And then so we're not completely confident that they were individual clones. So we can revisit that if you have a better protocol in your lab for further isolating the cells. So yes.
Perhaps that can be discussed at the end.
Yes, yes.
It's a good question. So
it is a
While
good
we were
question.
here, I was just seeing if the data was already sort of there or not.
Yes, we would share it with you, of course. Thank you. Okay, so then moving on over to the transgenic gene targeting mouse facility, which I'm co-director of. So in this core, we focus on the production of the genetically modified mouse lines. So while the MGD core is more molecular, in this core, this is where we perform the electroporation and micro injection and all the steps required to actually make the mouse. We also perform production of embryonic stem cell lines and other basic mouse services, such as cryopreservation of sperm and embryos and rederivation of mouse lines from blastocysts, frozen sperms, frozen embryos, and validation. This is a new service we're working on, is validation of transgene function and mouse fibroblasts. So both core, so what we use a lot is this CRISPR-Cas9 mediated gene editing. So I thought I'd just get everyone on the same page here. So we use a CRISPR RNP, which is a synthetic guide RNA complex with Cas9 to make targeted double-stranded breaks. And if you don't provide a repair template, the DNA is repaired 100% by non-homologous end joining, which is very error-prone and leads to random deletions and insertions and knocks out gene function. But if we provide a homology-directed repair donor, we can make a precise repair to generate targeted knock-in. So we've used these gene editing services to make null mutations and genomic deletions. So these are some of the models we either work with or make reagent for. And to knock in point mutations, make both small and large gene tags. I'm sorry, there's something with the timing. And also make conditional alleles, so CRE lines and LOX lines. And we've even done a few gene replacement models. So Diane alluded to this already. So it kind of what are efficiencies. So in the mouse core, we make about 24 lines, so 2 lines a month with 92% success rate. So what I mean by that is we get a positive founder that transmits to the germline. And then I kind of went through efficiencies by edit type. So we do, flocks to exons. So that's usually with a long single-stranded DNA. And these are kind of the percent of founders that are positive. Large protein tags, and we see much lower percentage here. And we also see lower germline transmission percentage for these as well. We've made large deletions in mice. And then we also make with the short oligo donors, we can do target SNP changes or small protein tags, which show pretty high efficiencies. So today, I wanted to talk about how we're working on improving these services. So first, I want to talk about decreasing the cost and timeline when we're doing those short genetic modifications, since they are quite efficient. Working to increase insertion efficiencies of long DNA sequences and generating embryonic lethal edits. For instance, if your SNP of interest when homozygous is lethal, that can make generating live embryos difficult, or when you're working on editing developmentally required genes, you can cause a lot of lethality as well. So first, to kind of get at how reducing cost and the timeline, I thought I'd kind of go over, well, what is the process? So you just contact us with your goals. We provide the reagents, then we validate them in mouse embryos. After they're validated, we design and synthesize the donor, introduce them, detect the target edit, breed those animals, detect it again, and then sequence. But for short edits, we've started to skip this validation stage because we've been doing this a long time. We have a lot of designing programs and experience. So usually our CRISPR reagents do work well. So we have been skipping that step, which saves time and money. And we just design and synthesize multiple donors. And then the other change is we sequence earlier to know how it worked, and we can actually predict better in mosaic animals using a technique called titer sequencing, whether it worked as well. And this, while we're on this side, this step 3, we do not skip that for large insertions because, as I'll go over later, these donor molecules for a long insertion are much more expensive, and they very much depend on where you're cutting. So we want to make sure that the CRISPR cutting is at a very high efficiency, since overall these edits are lower efficiency, and often we're using 2 CRISPRs for this, and we want them to be paired efficiency. So we still do quality control for those. So now what we do, so we just generate multiple guide and oligo donor combinations because they're relatively cheap. An oligo donor is only $150 to $200. And we'll electroporate smaller groups of mice with these multiple conditions, sequence the founders to select breeders and to make sure we have mice with the correct edits, and then generate the N1 generation and sequence verify with clonal sequencing at the end. So what is titer sequencing? So it's tracking of insertion, deletions, or recombination events using basic Sanger sequencing. So all we do is around your loci of interest, we sequence, we amplify that out from a wild type animal. And then we also have the reference here, which is the change we're trying to make. And that could just be a DNA sequencing file that you create. And then in this instance, the mixed pool would be your mosaic founder animal. And then we just submit them all for Sanger sequencing. And then there's 2 online programs for titer. In the output, it will predict what percentage of the sequence has deletion or insertion, which would be indel, and what percent of the alleles in this mixed population have the correct HDR you're targeting. And there's been studies comparing this technology to deep sequencing, and it is actually really comparable. So at getting the percent of HDR, it predicts that correctly. And then as far as the indels, it usually predicts correctly what percent are indels, but not correctly what they actually are. But we don't need that in our instance. And here's an example of that in action. I actually use this from a tool from Synthago. So this was a user's line, and this up top is the changes. They wanted these 3 changes, and then it predicts what alleles you have in that animal. And here you see this animal is predicted that 38% of the alleles are the correct edit, and then this animal was higher at 57%. So sometimes we get lots of positive founders, like upwards of 10, which would be expensive to breed everyone. And if you're just guessing, you might randomly pick one that has a low percentage and then not get germline. So we can use this to narrow down which ones we breed. So by making these changes, we've made the cost about 16% cheaper for small insertions. It's quicker, shorter timeline, about 5 months to N1 generation, which in our past techniques, this would be more like 6 to 7 months. And then also multiple independent lines are generated. So oftentimes both CRISPR conditions work, but in some instances, when we do finally do the founder breeding, we find that the allele that went germline has a mutation. But when we're kind of hedging our bets like this with multiple independent lines, we usually end up with a correct line instead of having to go back and repeat the experiment. So that's all I have about kind of that modification. If anyone has any questions before I move on to making large insertions.
I don't see any in the chat, but people are welcome to unmute themselves and ask if they have a question. That's pretty good efficiency and an incredibly reasonable price.
Mm-hmm. So the next thing I want to talk about is increasing insertion efficiencies of long DNA sequences. So first, as you saw from the table, we have pretty good success rates, but we do have limitations. So for a lot of our projects, we use a long single-stranded DNA homology directed repair donor, and that's very restrictive of length. I have less than 2 kb up here, but once you get over 1.2 kb, it gets pretty difficult to actually make that type of donor. There's a lot of sequence constraints. If GC content is high or low, or there's a lot of repeats, you can't generate a long single-stranded donor. Yield is often low. You can purchase these from IDT, but they're very expensive, and they're also expensive to make in the lab. They require microinjection, which is not really a problem, but that takes more mice and more time when you're using microinjection over electroporation. And then, since we have this length and sequence constraints, when we encounter a project that has one or both of those issues, we'll use a plasmid DNA donor, and these have low efficiency, very long homology arms, which then that complicates the characterization. So making those genotyping PCRs much longer and more difficult, and it also requires microinjection. So one thing that we're moving towards, we just got the IRB approval, and we'll soon have the IOCOC approval, is using AAV donors to generate knock-in mice. So this technique is called CRISPR ready, because you can pair it with RNP electroporation. So you use a AAV donor to infect your cells, and AAV, they have a naturally occurring single-stranded genome. So single-stranded DNA inserts more efficiently than double-stranded. The capacity can be up to 4.9 kb. That's another improvement. And it's doesn't require microinjection, so it's easier to make a lot of embryos at once. So you disinfect, then electroporate, and then transfer back into the mouse and generate your line. So there's a lot of advantages to AAV. So they very robust. There's two serotypes that very robustly transduce mouse embryos. So that helps increase editing efficiencies because you're getting a lot more of your donor in there. It's compatible with RNA or RNP electroporation, making the technique easier and quicker. And you can insert complex DNA sequences up to 4.9 kb, and it's much less expensive than long single-stranded donors because you actually only need a very, very small prep. And then another method that we're currently using to increase our efficiency. So we have some animals that we just generated with 2-cell injections, but haven't gotten to analyze it yet. But most injections that we do and other labs and cores do is at the 1-cell stage. But we've recently have been able to start doing 2-cell injections, and this has a couple advantages. So during 2 cells, that's when embryos undergo their major zygotic gene activation. So because of this, it means the chromatin state is much more open, so that can help increase editing efficiency. And also HDR, or homologous recombination, occurs during the G2 phase, and the G2 phase is much longer during the 2-cell phase of the embryo. And then I'll talk about this kind of in the next point. Another advantage of 2-cell stage is you can target just a single cell, and then that leads to more mosaicism. So not every cell in the animal is edited. And then if you're targeting a lethal gene, this can be very important for the survival of your founder embryo. So I was going to go on to the last point that we're working on, which is generating embryonic lethal edits or edits in genes that are required for development. So what kind of lethality issues are there? So first, a problem in CRISPR editing field that we all struggle with is that the error-prone non-homology end joining is the most efficient repair pathway following double-stranded breaks. Another issue we run into when we're making mice is we can observe very high homology-directed repair, but in some instances, the mutation we're trying to generate is lethal when homozygous. So we essentially, in the founder animal, we generate what looks like a homozygous animal. And also HDR can be imprecise, like shifting the reading frame, and that can also be lethal. So how do we deal with it? Well, one thing we can work towards is decreasing indel formation. Another thing is promoting heterozygosity of that mutation that you're trying to target, or just increasing mosaicism in the animal. So one way, so this that we're currently testing, so is adding dead Cas9. So dead Cas9 is catalytically inactive Cas9, so it still will recognize the guide RNA and complex with your target DNA, but it won't cause cutting. So the whole point is perhaps if you have dead Cas9 in your mix at a percentage of alleles, you'll block the HDR or indel formation. And this is from, these graphs here are from work done in mouse embryonic stem cells. So it hasn't been published or shown in embryos yet, which is what we're working on. So on the bar on the left of each graph is showing what proportion of edits are the non-specific, non-homologous end joining, or these are actually cell clones, excuse me, wild type, or have homozygous edits or heterozygous edits. And if they add a Cas9 or dead Cas9 with the Cas9 in your RNP mixture at a 1-to-1 ratio, I think they tested up to 1-to-2, you can actually increase incidences of hets. So perhaps, and also of wild type. So in our case, we can hopefully drive more heterozygosity and more mosaicism to get better survivability. And then the last thing I want to talk about is prime editing. So this has been on my mind for quite a long time, since it came out in 2019. But there's been a lot of work kind of perfecting the reagents and kind of getting formats that are amenable to use in all organisms. So what is prime editing? So prime editing is basically targeted editing without double-stranded breaks. And if you don't make double-stranded breaks, you're not going to see that non-homologous end joining, which can be very deleterious and lethal. So it consists of the prime editor protein, which is a Cas9 nickase. So instead of a double-stranded break, it just snips one strand, and it's fused to a reverse transcriptase domain, which helps incorporate your edit. It uses a different kind of guide RNA called PEG RNA. PEG RNA not only specifies where you're targeting in the genome, but it also contains the edit. So when you make the clip here, you get this little 3' flap, and the PEG RNA has a domain that recognizes and hybridizes to that domain. And then it has a reverse transcriptase domain that contains the edit. So you see it getting incorporated here. And then once that 3' edit is made as part of the repair process, the DNA undergoes flap equilibration, where this other flap's edited, or excuse me, degraded, and then the edit is incorporated. And down here they're showing, so up top, this is basically prime editing version 1. So people have been working to optimize these prime editor proteins. And in prime editing 3 and 5 systems, you actually introduce another guide RNA to nick the complementary strand, which helps increase efficiency. And then a newer advancement in these systems, they call these PE Max, is they express this protein here transiently that blocks mismatch repair, which can change the edit back to wild type. So that also increases efficiency. So prime editing has been used in mouse before, but it's very low efficiencies. And every time I'm at a meeting talking to another core lab, it just doesn't work great, but it would be good to get this up and running. So one thing that we're working towards, we have a collaborator that sent us some prime editing 5 Max and engineered PEG RNAs, which have this motif for stability. So we're hoping with these improvements that maybe we can get prime editing to work more efficiently in mouse embryos for our needs. So just to kind of summarize, I talked about decreasing cost and timeline by using multiple CRISPR combos and early founder sequencing, increasing efficiencies using long DNA sequences using AAV donors, which are coming soon, and 2-cell injections, which we're currently doing. And then working on better methods to deal with lethal edits and developmental genes. So right now we're testing the addition of dead Cas9 to our electroporation mix. And so far we've been able to increase survivability, but we haven't gotten editing rates, which we're happy with. And then hopefully in the future, prime editing. And then I have also listed the 2-cell injection for trying to work with lethal edits, because I mentioned you can make more mosaic animals if you just target one cell. So with that, I would like to acknowledge my crew. So in the MGD core, I have talented technician Lillian, and then several people. So co-director Kyle in the transgenic core, and her launch, she's our mouse microinjectionist, and Nick, our technician, my faculty advisory committee members that kind of have for both cores mixed in there, the University of Utah Research Facility Administration, and the other cores that I work with to make this work possible and pass funding for the MGD core. And with that, I'll take any questions.
There's a question in the chat. Do you want to go ahead and ask that? I'm gonna, Gunjan?
Yeah, sure. Am I audible?
Yeah.
So how much time it takes to generate a mutant cell line by CRISPR? It is very dependent on your cell line. So I have worked with lines that we successfully generated at 6 to 8 weeks. And then poor, like Mikkel was one of those ones where it drug on for, was it 4 or 6 months because of the cloning issue. So it just really is very dependent on cell type.
Okay, yeah, thank you.
Anybody else? Hang on, because we've got another presentation coming up about our metabolite protein core. Anybody else have a question? Otherwise, we will let Crystal unshare. Thank you, Crystal. And any questions you have, feel free to reach out to her at her core and she can help you. I can testify, at least for cell lines, that it has worked really well for me. Okay, and our next presenter is Kevin Hicks, and I'm not going to say any more. I want Kevin to have that time. We may go a little bit over just so people know, but stay on. And we are recording these and they will be available online in like a day or two. Okay, Kevin.
Great. Can everyone hear me?
Yes.
Awesome. Perfect. Well, hey, Diane, thanks for that introduction and the opportunity to present everybody. And yeah, thank you all for joining in today. So I'm a research instructor in the Department of Biochemistry, and I work very closely with Jared Rutter's group. And today I'll be telling you about new technologies in protein metabolite discovery. So I co-direct the Utah CIHD Protein Metabolite Discovery Core with James Cox. So what I'm going to tell you about kind of the rationale for, this realm of biology, tell you about services of the core, give some examples of discoveries we made, and then go into some of the newer kind of innovations that we're going to take this platform and provide as new services. Oh, there we go. Okay, so the protein metabolite interactome is relatively unexplored, right? If you compare it to other interactomes in biology, right? So this schematic here just kind of outlines some of the different transfer of information through the different domains of biology, right? And where my research program is focused is really on the interactions between small molecule metabolites and proteins in the proteome, right? Traditionally, this has been a pretty hard area to systematically interrogate, simply because the technologies just weren't there and haven't kept up with other omics platforms. So that's, really one of my main research goals, is developing new technologies to tap into this realm, right? And, you could discover substrates, products, cofactors, but mainly, I think my main interest is discovering new orthosteric and allosteric regulators, right? So small molecules that regulate protein function. And we believe that, this metabolome orchestrates broadly proteome function, not just within metabolism, but across various cellular pathways. And then beyond just kind of the fundamental knowledge of how the biological network is organized, you could then also take this information and potentially use it for drug discovery, right? So you have a known metabolite binding a target protein of interest, and if you know where it binds, then you can theoretically drug that site. So that's just kind of the application side of it. Really, I also wanted to kind of convey this in another kind of format, right? So what you're seeing here, this is the kind of the canonical metabolic network as defined by KEGG. So the Kyoto Encyclopedia of Metabolism, essentially. And it's a two-dimensional image, right? So each dot or each point is a small molecule metabolite, right? And then the line between the dots is an enzymatic reaction, right? So this is just displaying substrate to product conversions, right? But there's a whole nother layer of interactions on top of this that we really don't know, right? For example, right, so this is the last step in glycolysis, pyruvate kinase, isoform M2, right? So it takes phosphorylyl pyruvate to pyruvate before entering the TCA cycle, right? So just for this one enzyme, there's a very well-characterized allosteric activator, that's fructose 1,6-bisphosphate, which is an intrapathway activation. So F16BP is upstream of pyruvate kinase M2, right? But there are also many other interpathway allosteric regulation. So we have L-serine, which is an amino acid that activates pyruvate kinase. Phenylalanine, another amino acid, inhibits pyruvate kinase. And then a very distal interaction, so SACAR, it's an intermediate purine nucleotide biosynthesis, is an activator of pyruvate kinase, right? So for just this one enzyme, and I'm not even displaying all the other known regulators, there's about a dozen different small molecule metabolites that coordinate pyruvate kinase function, right? So then if you extrapolate from 0 to about a dozen or so for this one protein across the network, there are probably hundreds of other regulatory interactions just within metabolism between metabolites, metabolic intermediates, and diverse enzymes, right? But that's just metabolism. If you then look across the entire proteome, I believe that there's going to be thousands of these interactions coordinating cellular processes, growth factor signaling, immune pathways, developmental pathways, et cetera. And there are examples of these sorts of interactions, but not at a systematic level. That kind of transitions to our technology. So while in Jared's lab, I developed this platform known as MIDAS. And at its core, it's based on a classic biochemical technique called equilibrium dialysis, right? So we take our highly concentrated, highly purified target protein of interest, separated by a defined cocktail of metabolites. So we have a custom metabolite library. I'll talk about that in a second. The previous library was 401 metabolites, right? So separating these two chambers is a 10 molecular weight cutoff dialysis membrane. So essentially your target protein is trapped in the top chamber, but your metabolite cocktail can freely diffuse across both chambers. So we set that up, allow it to equilibrate. If one or more of those metabolites in the cocktail interact with the target protein, they can be enriched in the presence of the protein, so in the protein chamber, and or depleted depending on the nature of the interaction. So we'll have, so for those metabolites that do not interact with the protein, they're essentially equal on both sides. We then crash out and denature the protein to remove it, and then analyze the relative abundance of all metabolites in parallel in the top chamber and the bottom chamber, using either a high throughput flow injection mass spec technique, or where we're moving to now is a more conventional LC-MS metabolomics platform. And I'll kind of go into that a little bit later. But what you end up with are kind of three different scenarios. So metabolites that passively interact with the target protein can be enriched in the protein chamber relative to the metabolite chamber. So we'll see a fold enrichment of that interacting metabolite. Those metabolites that don't interact are equal in both chambers. So their peak intensity would be the same. In some scenarios, we'll actually see a depletion of a metabolite, and that could be due to an enzymatic consumption. So that target protein is converting a substrate to a product faster than the diffusion rate. And in some circumstances, these metabolites can covalently attach to the target protein. So when we remove the protein, we essentially pull that metabolite out. And in those scenarios, we'll see a depletion of the metabolite in the target protein chamber. So that's the general MIDAS platform. Where we were at, this was all soluble proteins and using a custom metabolite library. So as I said before, the previous library, this was as of about 6 months ago, we're kind of on the end of that library, was 401 metabolites. This is just the chemical taxonomy distribution of the library. And then looking at human metabolism overlaid on the KEGG metabolic map. So all of the green pathways are human metabolic pathways. The red dots are the metabolites in that 401 MIDAS library. Where we're at now, and this has been quite an effort, but we finally have the complete library together. We're up to 736 metabolites. So we've almost doubled the size of the library and increased coverage of core and kind of ancillary human metabolism. So that should allow essentially deeper discovery per target protein. So this is just one example of using the platform. To date, we've screened over 230 proteins using that 400 compound library. Again, we're going to be moving everything into the 730 compound library. But previously, we screened 3 proteins known to regulate the mTORC1 growth factor signaling pathway. So we screened Castor1, Cestrin2, and Reb. And these were essentially, well, both positive controls and maybe trying to discover something new. But the main reason we targeted them is that they are known metabolite interactors. So arginine inhibits Castor1, binds it allosterically. Castor1 is not an enzyme. Leucine, isoleucine, and methionine interact with Cestrin2 and functionally regulated, again, non-enzymatically. And then Reb is a slow GTPase, so it interacts with GTP and the product GDP. So we put all 3 of those through the MIDAS platform, and lo and behold, we found basically what you'd expect. So castor 1, highly enriched and highly significant arginine. Cestrin 2 enriched both all of the leucine and isoleucine and allo-isoleucine isoforms, and then also methionine. And then Reb, we saw both GTP and GDP, and then also related metabolites, right? So we also detected an interaction with ATP and deoxy GTP, so derivatives of the known substrates. And this is something we commonly see within our MIDAS data sets, is you'll get either a known or novel metabolite, but then you'll also get structurally related derivatives of that metabolite. So if you pull out NAD, for example, you might get nicotinamide and other dinucleotide derivatives. But this just served as a nice proof of principle that the platform is robust and it can detect both enzymatic and non-enzymatic protein metabolite interactions.
Hi, Kevin. I have a question.
Yeah.
So these metabolites are known to regulate the protein activity. So how do you decide the metabolite concentration in the chamber? That how much metabolite to keep in the chambers so that they are not either regulating or modifying the enzyme activity? Or do you change the metabolite concentration in the chamber?
Yeah, so let me go back. There we go. So I didn't go into all the details here. So the original platform, we had all the metabolites at 50 micromolar. So essentially they're all equal. And the idea there is to basically give all of them equal competition on the target protein, right? What we saw more often than not was that even, again, it kind of comes back to that kind of substructure similarity, is that you can, even within that screen, we can pull out the primary ligand and then lower affinity ligands that share similar structure, right? 50 micromolar here, that was a previous platform. I'm not going to go into all the details, but where we're moving to now with the new LCMS platform is a much lower concentration. So we'll be screening at about 10-fold lower. So essentially, when you look at the stoichiometry of moles of binding site per moles of metabolite, it'll be in vast favor of the binding sites.
Okay, thanks.
Yep. Cool. Yep. So let me go back here. So, yep. So the mTORC1 regulators just served as a nice proof of principle that we can discover known interactions. There's other things we haven't even followed up here. So castor and cestrin 2 both also interacted with polyamines, and we're not really following up on that, but that's back in the hands of our collaborators at this point. Okay, cool. So what do we need for MIDAS? What are our sample requirements? So for the new platform, again, where we're moving to, we'll need 300 microliters of 200 to 500 micromolar target protein. So this protein concentration is in the realm of what you might be using for a crystallography trial, right? Now, there is some flexibility there, and just reach out to me if you're kind of on the low end of that or below, like you can't get your target protein to high enough concentration. And there's things that we can do on our side to compensate for that, but it's really just on a case-by-case basis. So high concentration of protein, and then high purity. So if there are co-contaminants, especially if they're enzymes, we could then, potentially wander into false positive discovery. So it has to be pretty pure. Again, kind of in the same realm of what you'd want for, X-ray crystallography. And then our cutoff right now is 10 molecular weight for the dialysis membrane. So usually we ask for our targets to be above 15 kilodaltons. With that said, we have screened lower because we can get our dialysis plates with smaller dialysis membranes or the pore size. So protein buffer, we ask that your target protein is in 25 millimolar heaps at neutral pH in 100 millimolar sodium chloride, pretty standard protein biochemistry buffer. If your specific protein requires reducing agents, detergents, metals, other additives, again, that'd be a scenario where you just contact me and then we can kind of hash out what will and what will not work. I can say that in most scenarios, especially in our new LCMS platform, if other additives are at or below 1 millimolar, they don't, at least from what we've seen with detergents and metals and reducing agents, they don't perturb our ability or our chromatography, so we can still get good detection of metabolites. So that's what we would need for the new kind of MIDAS approach. So what does our core do? So kind of the bread and butter is this new technology, the protein, MIDAS, protein metabolite interactions. I'm not going to talk about it much today, but James Cox and the proteomics core can do non-enzymatic or detect non-enzymatic covalent modifications. So what we can do is screen your target protein, tell you what metabolites putatively interact with it, right? But then we can also take the protein fraction, the denatured protein, and then send that off for proteomics to ask the question, okay, of those metabolites that hit your target protein, do we potentially see them as covalent modifiers on your target protein? So we can kind of get a little bit deeper into the data set. Beyond kind of the discovery platforms, we also offer orthogonal validation and some characterization of your interactions. So kind of like our first line secondary screen, you could call it, is differential scanning fluorimetry. So this is a plate reader assay to essentially on a protein per metabolite basis, ask the question if that metabolite changes the melting properties of your protein. And we can do this very rapidly and with, dozens of metabolites in a very short order of time. But there are false negatives with this because it doesn't work for every protein. So it's really something we kind of have to optimize. But for a first pass optimization, it can get you a rough KD of the interaction. Kind of new what's coming up, we'll get this a new instrument at the end of February. This is I would say kind of on the cutting edge for characterization of macromolecule ligand or receptor ligand interaction measurements, and it's called a microscale thermophoresis. So again, it's another fluorometric technique where you have your receptor, your target protein, either labeled, chemically labeled with a fluor, or we can also use intrinsic tryptophan fluorescence. And then you do a dose titration of your ligand, and basically based on the fluorescent properties of the protein and its movement within this capillary, we can then develop a binding curve and get you a binding affinity from it. So this is a pretty new technology, but it's very robust and there's a lot of use of it, across multiple different fields. So I'll have that service up and running probably in the beginning of March. Okay, cool. Just a quick little vignette here. How am I doing on? Okay. Yeah, so while in Jared's lab, I developed the MIDAS platform and we screened, 230 plus proteins. I focused a lot of my efforts then on characterizing metabolite interactions within core human carbohydrate metabolism. So we use that kind of as the proof of principle of our platform. So of that 200 plus proteins, we screened 33 enzymes within glycolysis, gluconeogenesis, TCA cycle, and some of the branching pathways. And we discovered 805 significant protein metabolite interactions. That includes both known substrate product, regulators, etc., and novel interactions. Within that data set, what we observed was both extensive intra-pathway interactions. So these lines are basically showing interactions between a metabolite and a protein. So basically, metabolites from the same pathway coordinating or interacting with enzymes of the same pathway. But then also extensive inter-pathway interactions. So these would be diverse metabolic pathways converging on enzymes of human central carbohydrate metabolism. It's a very rich data set. Our manuscript is up on bioRxiv. We're in revision at Science right now. So hopefully this comes out soon. There's a ton of data in here. I don't have time to go through all of it. But if you are at all interested in carbohydrate metabolism, definitely have a look because there's way too much for one small group to follow up on.
Kevin, this is Diane. Just a quick question for your core services.
Yeah.
Do you guys provide help with that analysis?
Which analysis?
Well, I mean, making those big tree, it's sort of bioinformatics, making the trees of connections and things like that in the discoveries that are made.
We offer, so not this sort of pathway analysis here. So this would be pretty extensive. This is 33 enzymes, right?
Yeah.
So this would be a large project. But what we do offer is we have a portal, a graduate student in a computer science graduate student in Jared's lab made a tool that interfaces with MIDAS data. So you can take your target protein that we've screened and the data set generated from MIDAS, dump it into his tool called Electrum. It's on GitHub. And I believe it's in preparation to be published. And what that'll do is essentially, it's a nice way to peruse the data. So it's an interactive website. So you have your target protein, all of the metabolite interactions with that protein, right? Whether they're positive or negative enrichments, their statistic cutoff, but then it'll also link you out to many other online databases, right? So it's basically a way to kind of sleuth through your data and start to figure out what these connections might mean and generate hypotheses from it.
Perfect. Thank you very much.
So I'm just going to focus on a very kind of one of the vignettes from that manuscript, and that's some novel discoveries with lactate dehydrogenase. So this enzyme serves an important conduit in pyruvate metabolism. So pyruvate's at the end of glycolysis, feeds in the TCA cycle and other anabolic pathways. LDH is the only enzyme or isoform family that will convert pyruvate to lactate interconvertibly. So this is important for either dumping lactate out of the cell, like an exercising muscle, or taking up lactate as an anabolic fuel. And some of you have probably seen Josh Rabinowitz's work that shows that lactate is one of the major circulating substrates of carbon on an organismal level to feed biosynthetic pathways. So it's all served by LDH. It's an NADH dependent enzyme. There's another layer to that, in that there are multiple isoforms of LDH. So we focused on LDHA and LDHB, which are the dominant isoforms. And the kinetic properties of these isoforms kind of drives their reaction directionality. So LDHA is canonically viewed as primarily converting pyruvate to lactate, and LDHB is more biosynthetic, taking lactate to pyruvate. These enzymes share 75% sequence identity, so they are highly similar, and they're above 85% just on residue similarity. So they're very highly similar. We put these through the MIDAS platform. We saw very similar hits on both LDH-A and B. So nicotinamides, small keto acids, nucleotides, et cetera. If you kind of take a bioinformatic approach, you can then class these different metabolites into families. So again, we have nicotinamides and dinucleotides here in purple. Nucleotides, so ADP, AMP, deoxy-GDP, et cetera, as another purine nucleotide hit family. We saw small keto acids similar to lactate and pyruvate. Oxaloacetate is a known endogenous inhibitor of LDH. And then one that stood out to us was we saw a couple different CoA and acyl CoA derivatives as hits on both LDH A and B. And that's completely novel. So if we have substrates, right, nucleotide, that's a whole nother story that's pretty interesting, but I don't have time. And then these acyl CoAs. So one of the major uses of acyl CoAs in the cell is as fatty acid intermediates. So for either beta oxidation or de novo lipid generation, et cetera. The major, the hub acyl CoA is palmitoyl CoA. So we purified LDH-A and LDH-B, and then performed enzyme kinetic assays and asked the question, okay, if we go from CoA and then short to long acyl CoAs species, what does that do to enzyme activity? And what we found is that short acyl CoAs didn't do anything on either LDHA or LDHB. But then when you got to above, above 12 carbon acyl chain, we started to see inhibition of LDHA. And this really, I would say kind of the most potency started around palmitoyl-CoA at the C16 acyl-CoA length. And there we had an IC50 of around 1.5 micromolar. So long chain acyl-CoAs appear to inhibit LDHA at, near, endogenous levels, whereas LDHB was completely insensitive. It didn't matter how long the acyl chain was or how high in concentration, LDHB was insensitive. So that was all in vitro kinetic assays. What about in cells? So we developed two different tracing regimes. So we took 13C glucose, fed that, or 13C lactate, right? So we got two different directionalities through the LDH reaction and fed that to H9C2 cells with or without palmitate treatment. So essentially we're tracing through glycolysis with glucose to pyruvate and then measuring extracellular lactate, or we're feeding lactate and then measuring intracellular pyruvate. So we're kind of isolating the LDH reaction. And then in collaboration with Crystal Davey's core and Bai Lo's core here at the University of Utah cores, we generated LDHA-B or LDHA-B knockouts within this H9C2 cell line. And what we saw is that if you look at wild type, you treat with palmitate, we see decreased heavy carbon labeling of lactate, extracellular lactate, or intracellular pyruvate. So suggestive of, palmitate can inhibit this reaction, right? But we further isolated that. So if you knock out LDHA, we no longer saw that inhibition. So the cells are essentially reliant on insensitive LDHB. If it's only LDHB present, you still see that inhibition. And then if you have both of them knocked out, essentially you're at really low substrate, pyruvate or lactate, and you no longer see that sensitivity. So it really showed that within, in this cell line, that LDHA is sensitive to palmitate inhibition, where LDHB is not. So basically our model for this is under low fatty acid or kind of basal fatty acid levels. LDHA and B are both active. They can interconvert pyruvate to lactate. Pyruvate can enter the TCA cycle. Now, under fasted states or high fatty acid states, long chain acyl-CoAs will go up in the cell. That will inhibit LDHA, preventing pyruvate to lactate conversion, right? Palmitate can feed the TCA cycle, essentially sparing carbon from pyruvate, right? And then that will allow pyruvate to either, let's say in the liver, enter into gluconeogenesis or feed into other anabolic pathways, nucleotide metabolism, amino acid, et cetera, right? So it's essentially a way for the cells to make a decision of what carbons they're using for energy, right? Is it glucose or is it fatty acids? And we're following this up further, primarily in liver models, but we're also looking at it in the heart as well. But that's for another day. Okay, real briefly, I'm going to talk about some of the new technologies that we're developing. This is pretty preliminary, but we are laying the groundwork now. And I really hope that some of you are interested in it and maybe want to collaborate with us and put your proteins through. So the first one is membrane proteins, right? So current MIDAS can only target soluble proteins, right? Now, that's great. That's about 61% of the proteome is soluble proteins, but there's another 23% or so, which is membrane proteins, right? We want to be able to target that, fraction of the proteome, right? Another reason for that is if you just looked at FDA-approved drugs, right? More than 50% of FDA-approved drugs are targeting membrane proteins, right? So even though they represent only 23% of the proteome, they represent more than half of, drug targets, right? So, and there comes in kind of some of the application of MIDAS for revealing, novel drug targets, right? So, right now we're developing the new MIDAS platform. This is in collaboration with Calico Life Sciences in the Bay Area. And our pilot model is GPCRs, right? So the platform is very similar to what I described before. So it's still based on equilibrium dialysis, right? Now, there's obviously some major differences. The first one is your target protein is either going to be in a lipid nanodisk or detergent bound micelle, right? So right now we're actually optimizing the platform for detergents because more typically membrane proteins, you can get a higher yield of detergent membrane proteins rather than the nanodisk format. But either route should be amenable through for this Midas analysis, right? So you have your target protein in your detergent vesicle, you perform the dialysis as before, right? And then the major difference is this downstream. As I mentioned before, we're moving to an LC-MS metabolomics platform, and that's really to allow for more non-volatile buffer components, so heaps, other buffering components, salt, right? But then also the detergents, right, or phospholipids that would come along with your membrane protein, right? So I don't have a whole lot of data for you right now, and I didn't want to take you through all the method development, which is kind of dry. But I'll tell you right now that the first pilot protein is going on the mass spec today. We have the adenosine A2A GPCR up for analysis. So unfortunately, I can't share the data with you yet, but that is going to serve as kind of our first proof of principle pilot to see if we see adenosine as an enriched interaction. So if you have membrane proteins as part of your research portfolio and you want to know what metabolites might interact with them, we're going to have the platform up and ready here probably in the next month. The other direction we're taking this is instead of using a rather expensive custom metabolite library, we're going to tweak the platform so that you can use your favorite cell lysate or tissue metabolome, right? So again, very similar. It's all based on equilibrium dialysis with subtle tweaks across it, but I'm not going to go into all the details. But instead of loading in our defined metabolite library, you can grow up your cell line, extract polar metabolites, and then we'll use that as the bait in the Midas platform, right? So to date, we've used this with, we're piloting it with 293 T cells, basically growing them up and then extracting the cell lysates. And then we've also done it with whole livers. So essentially mouse livers, freeze-dried, ground them up, extracted polar metabolites, and then use that as our, our screening metabolome. This has some very unique advantages and potential disadvantages relative to the custom library. So you have potentially the entire metabolome, right? So it's a much more non-targeted LC-MS metabolomic approach than the custom library, right? So you have whatever metabolites are within that cell at a given time that you can detect on the mass spec, right? So theoretically, it's much larger than a custom library, right? It also allows you to the non-target approach. So we may not even know what a unique mass is on the mass spec, but we can tell you if that mass is interacting with that target protein and then do the non-targeted workup, fragmentation, et cetera, to ID that mass, right? This also goes back to the question before, is that you'll have all metabolites at their endogenous concentrations, right? So, if, a ligand is at the appropriate concentration under your, cell line growth conditions, right? Relative to other, competitors, we could theoretically detect it, right? That also kind of brings in one of the caveats, right? Is that there are a lot of intermediate metabolites that are at vanishingly low concentration, right? So we may have them in our library and detect them in the other platform, but when you purify them from a cell, it could be so low that we don't get any signal, and thus it's kind of a false negative, right? But one of the coolest advantages here is A, it's much cheaper than the custom library, and B, you can then grow your cells however you want, different nutrient situations, fed versus fasted, etc. And then ask, for those metabolomes, what are the unique protein metabolite interactions? What we were doing with the livers too is basically going to work towards a model where we had healthy versus fatty livers, right? Extract their metabolomes and then ask, are there different protein metabolite interactions in the healthy versus disease states? So again, we're kind of working through all of this, so I don't have a whole lot of data to share, but it's really, it's hot on the press. And with that, I'll wrap up here. So there are way too many people to thank. This platform itself is super highly collaborative. So those 230 plus proteins that we've screened, the vast majority of them are from internal and external protein crystallography, biochemistry groups, et cetera, both academic and commercial. So there are a lot of people participating in this platform. The biggest shout out I got to give to is to my team, basically, right? So, well, first off, my mentor, Jared, who really, you know, while I was a postdoc with him, really helped me develop this platform. And then Andrea, my very talented technician who keeps the wet lab component rolling. Casey Hughes is doing all of the LC and flow injection mass spectrometry, so kind of the back end of the Midas platform. Aaron Leifer is a very talented graduate student following up on some of the LDH discoveries that we've made. Ajak Arak is an undergraduate working with our group, and he's following up on some other protein metabolite interactions that I don't have time to talk about today. And then I can't go without thanking James Cox, my mass spec mentor, for teaching me all of the nuanced techniques and how to handle mass spec failures, et cetera. So lots of people to thank. I got to thank Crystal, Davey, and Bilo for reagent generation. They made a lot. Crystal made a lot of the protein constructs we used for purification in tandem with Bilo to make our H9C2 LDHA knockout lines. Multiple members from the Rudder Lab, my funding, including the funding from the iron and heme disorders. And then also you can contact me either at CIHD Cores, we have a website up there, or feel free to just shoot me an email and we can discuss your project, your target protein, and what you might want to discover with it. So with that, I'd be happy to take any questions.
Thanks, Kevin. Anybody want to unmute and ask more questions? It's exciting to think that you're going to be able to do those membrane proteins if people have purified ones. Gunjan, do you have a question?
Yes,
Yeah.
I have a question. So when you try to overlap that Midas data with LCMS data of cellular metabolomics, so how much overlap do you see?
Yeah. So I think it's going to be cell type dependent. So where our pilot started was with the 293T cells, right? And what we were getting around from our library. So we had, I think we had about 250 total unique masses. And again, we're kind of still tweezing apart the right cell concentration to use for this. So 250 total masses that we had reliable signal for. Of that, about 75 of those metabolites were metabolites within our custom library, right? So I would say there's some overlap, but if I don't have a Venn diagram or anything like that, but there's still, I would say, much more unique masses within the non-targeted cell lysate Midas versus the more custom kind of metabolite library Midas. Mm-hmm.
Okay, thanks.
Mm-hmm.
Other questions? I don't see anything populated in the chat. I just want to encourage people to reach out to these two core directors if you have questions or interests. Also, remember from the beginning of the talk that if you go on the cceh.io website, there's opportunities to apply for funding where you might think of a project that would utilize either one of these cores, and you could potentially get funded and core credits at the protein metabolite core or the mutation generation detection core to sort of spearhead and start a new direction in your research or an idea that you have. So I highly encourage you to do that. And thank you for joining us. This is recorded and will be made available on the CIHD website within the next day or two. So if you need more information than missed some of this, you can go there and watch it again, or watch parts of it that you want to. And I hope this has been helpful for you. And thanks for joining. Any other questions? So Mikhail, just, reach out to Crystal there. And I know
Yeah,
that.
I'm going to.
Yeah, you guys maybe
fine.
should set up a Zoom or something like that to kind of do the details so that we don't hold on to other people's time at this point. But definitely reach out because I know she can help you with getting that sequencing information for what's going on.
Thanks.
Okay. Thanks again for the presentation and thanks for joining us. And we'll see you next month again.
Thanks, Diane.
All right, take care.
Bye.
Bye.